Plasmids represent the extra chromosomal genetic material or DNA segment capable of autonomous replication in cells. They are found in most bacterial species. Most plasmids are circular, double-stranded DNA molecules that are isolated as supercoiled molecules. However, other forms such as linear double-stranded molecules have also been described. Plasmids are not confined to bacteria. They have been isolated from yeast, protozoa, and plants. In humans, certain viruses, including human papillomavirus and Epstein-Barr virus, can also exist as plasmids within cell nuclei. In this form, the virus replicates along with the nuclear chromosomes. The molecular biology of plasmids has been most intensively studied in bacteria. Certain bacterial plasmids have the ability to transfer themselves physically from one cell to another, thereby spreading through a population. Special properties of certain plasmids even permit interaction between two bacterial cells of different species. Special properties of certain plasmids even permit interaction between two bacterial cells of different species. These transferabilities are central to the role that plasmids play in bacterial evolution. The genes carried by plasmids represent a mobile reservoir of genetic information that can elbow the host bacterial cell with the ability to grow in otherwise hostile environments. An addition of intermediate relevance to humans is that bacterial antibiotic resistance is often conferred on bacterial cells by plasmid genes. Certain plasmids are capable of efficient transfer to other antibiotic sensitive cells, resulting in rapid horizontal conversion of an entire bacterial population to drug resistance. Moreover, it is common for a single plasmid to confer resistance to multiple antibiotics. Bacterial plasmids span a wide range of sizes, from those containing one or two genes, several kb in size, to those carrying several hundred genes, more than 500 kb. Because certain plasmids are so large and capable of carrying so many genes, it is possible for them to generally alter the metabolic capabilities of their host cell. In the absence of selective pressure, however, having the plasmid present is irrelevant to the host, and the plasmid is genetically dispensable. Under these circumstances, the plasmid could conceivably represent a genetic burden to the cell and would therefore likely to be lost from populations. The evolutionary mission of the plasmid, however, is to propagate itself and ensure its own stable transmission in the population. Despite being nothing more than naked DNA molecule incapable of surviving outside the cell, bacterial plasmids have evolved quite sophisticated mechanisms to ensure their own survival. An important property of bacterial plasmids is that they exist in characteristic copy numbers per cell. A central problem faced by all plasmids is the need to coordinate their replication with that of the host chromosome so that they are not diluted by cell reproduction. Bacteria are capable of reproducing at widely different rates. In the lower intestine of mammal, bacteria might divide once per 24 hours. In a rich medium in the culture flask, they can divide every 20 minutes. Bacterial plasmids must be able to sense the division rates of their hosts and alter their own replication rates accordingly. So, plasmid replication must also be regulated so that it does not compromise host chromosome replication and the life of the cell. Since plasmids are stably inherited DNA molecules, they have served as valuable research models to investigate the molecular mechanisms that control DNA replication. Because of their small size and genetic distensibility, they are easy to manipulate with the techniques of recombinant DNA technology. As we will describe later, plasmids have played a key role in the development and use of recombinant DNA technology. In this chapter, we will briefly describe some of the central features of plasmids, concentrating on bacterial plasmids because these are most widely understood at present. We will focus on the replication properties of plasmids and the important property of plasmid transfer. So, plasmid borne genes. Bacterial plasmids play an important role in human disease. This was illustrated dramatically by the discovery during the 1960s of bacterial strains that had acquired simultaneous resistance to four antibiotics in heavy use in Japanese hospitals. The multiple drug-resistant traits were transferable to sensitive strains and were responsible for the epidemic of bacterial dysentery. The antibiotic-resistant bacteria harbored a large plasmid called R factor for resistance, carrying genes that encoded proteins that destroyed or inactivated the antibiotics. Other R plasmids have been discovered that render bacteria resistant to heavy metal found in the environment, such as mercury and lead. Other R plasmids have been discovered that render bacteria resistant to heavy metals found in the environment, such as mercury and lead. The presence of the plasmids in bacteria allows for bacterial growth in heavily contaminated environments. A bacterial cell's ability to resist the action of an antibiotic to neutralize toxic environmental substances usually depends upon the production of large amounts of few key enzymes. These enzymes act by attacking the harmful substance. By virtue of encoding these key enzymes on plasmids, which function essentially as mini chromosomes, the bacterium is well suited to produce large amounts of required enzymes quickly because the plasmid is often present in multiple copies. With multiple copies of the relevant genes, transcription quickly generates sufficient enzyme to combat the antibiotic or chemical challenge the bacterium faces. E. coli is a normal resident of the human large intestine. Certain plasmids encode endo E. coli with the ability to grow in new environments and thereby convert an otherwise harmless bacterial resident into a pathogen. Such plasmids encode proteins that can alter the bacterial cell surface, permitting addition of the bacteria to the lining of the small intestine. A second gene, sometimes present on plasmid, encode as to secrete a protein toxin called enterotoxin, which can damage intestinal cells and thereby responsible for symptoms of dysentery. Other plasmids, called cold plasmids, provide a selective growth advantage to cells carrying them because the plasmid encodes a secreted antibacterial protein called a colicin that kills cells lacking the plasmid. Cold plasmids provide a selective growth advantage to cells carrying them because the plasmid encodes a secreted antibacterial protein called a colicin that kills cells lacking the plasmid. Immunity to the effects of the colicin is conferred on the host cell by a special protein also encoded by the plasmid. The mechanisms by which colicin proteins act to kill cells are varied. Certain types, such as colicin E1, produces holes in the membranes of sensitive cells, permitting ions to flow out. Others, such as colicin E3, enter cells and specifically cleave ribosomal RNA, blocking protein synthesis. Then plasmids can provide advantage to their hosts by other means, such as carrying DNA restriction or modifying genes. These genes encode sequence-specific DNA binding proteins that recognize and degrade DNA which is foreign to the cell. While physically altering the host DNA so that it is not degraded. Modification happening. Several hundred distinct restriction modification systems have been described in bacteria. Their natural function is to protect the host bacterium from insertion by an unwanted virus. In certain instances, the viral DNA is chemically modified so that its ability to replicate is impaired. In other cases, the DNA of the infecting virus is destroyed. This short circuiting of the phage replication cycle. Destruction of the virus DNA is achieved by specific nuclease enzymes. Those enzymes recognize specific nucleotide sequence and cleave DNA at those sites. These systems are the sources of restriction in the nuclease, which are essential molecular tools for recombinant DNA technology. Many of these restriction modification systems are plasmid bound, which others are carried in the host genome. Colicin secreted protein kills bacteria lacking plasmid that encode colicin immunity protein. Enterotoxin secreted protein alters ion balance of eukaryotic cells responsible for water loss from cells. Then plasmid transfer. There has been speculation that bacterial viruses evolved from plasmids. Small fragments of bacterial cells' chromosome may have existed in the form of primitive plasmids, which eventually evolved into complete bacteriophage. The ability to transfer cells from one bacterial cell to another, a property possessed both by plasmids and viruses, no doubt contribute to evolution of bacterial species. From this evolutionary perspective, perhaps no plasmid-bound functions are more important than those permitting plasmid transfer from cell to cell, thereby mediating the flow of genes through a bacterial population. This process of plasmid transfer is called conjugation. Conjugative plasmids carry a large block of transfer genes responsible for the specialized cell structure and enzymes required to physically move the plasmid genome from a donor cell to a recipient. Such plasmids are said to be self-transmissible. The phenotype conjugative plasmid E. coli is the F plasmid or 6 factor, a large circular plasmid of approximately 95 kb present in 1-2 copies per cell. Cells carrying the F plasmids are called males and as capable of mating with cells lacking the F plasmid females. So cells lacking the F plasmid are often represented by the notation of F negative, while F positive is used to represent cells having an F plasmid. Conjugation is a complex process involving the gene products of a large gene complex within the plasmid called the TRA or transfer operon. TRA for transfer operon, consisting of more than 20 genes. An operon is a set of contiguous genes expressed through a largely polycystonic mRNA, and male cells are mixed with female cells. Mating pairs form, joining together by a special structure called conjugation bridge. Certain F genes encode proteins for specialized male surface structures called F-pilus. The F-pilus is essentially a tubular extension of the bacterial cell surface, probably interacts with specific receptors on the female outer membrane surface to establish a conjugation bridge. Following cell-cell interaction, one strand of the supercoiled F DNA is nicked at a specific sequence called ORIT. This is accomplished by special DNA endonuclease encoded by the F plasmid. Other proteins encoded by TRA then bind to the exposed 5' terminus and guide that DNA strand into the female cell. As the DNA strand enters the female cell, it is replicated coordinately with the other plasmid parent strand remaining in the donor. Following DNA replication in both donor and recipient cells to convert the single strand into double-stranded DNA, a new F plasmid are circularized. The closed circular DNA is then converted to a supercoiled form by the enzyme DNA gyrase. The process is very important. The result of conjugation is the semi-conservative replication of F in both cells. Initially, a female daughter cell is converted into a male cell by this process. It then is capable of transferring the F plasmid. Thus, the F plasmid can be thought of an infectious agent capable of rapid spread through a population of female cells. The ability to mediate conjugation is not confined to the F plasmid. Other plasmids, particularly the R plasmids, also carry the genetic information required for conjugation. When these plasmids are transferred, the female recipient becomes resistant to antibiotics, which can be inactivated by the products of certain R plasmid genes. As the F plasmid transfer, the recipient female becomes a donor male capable of subsequent transfer. Plasmids such as whole even, mentioned later, are not self-transmissible by themselves. They have, however, evolved the ability to take advantage of the genetic free ride offered by co-residing conjugative plasmid. When the conjugative plasmid is transferred, smaller plasmids such as co-even can also move into the recipient and are said to be mobilizable. So F plus male and F negative female, cell-cell contact. Host chromosomes are not shown to simplify understanding plasmid behavior. The nicking of F plasmid DNA at ORIT, attachment of proteins to free 5' terminus, initiation of DNA synthesis from the free 3' OH in donor cell to generate rolling circle replication intermediate, entry of 5' end of F into female. Then continued replication in donor cell, initiation of DNA replication in recipient cell. Then completion of replication in donor cell, cleavage of transferred strand from replication intermediate, formation of two complete circular F DNA molecules, F positive male and F positive male 2. produced 3' and starts the replication. So, a model for transfer of F plasmid DNA from F positive male donor cell to F negative female recipient cell during a rolling circle replication mechanism for the type described. F plasmids can transfer chromosomal genes by recombining with the host genome. Conjugation, as previously described, is genetically manifested only through the conversion of the recipient cell into a donor. However, conjugation was initially discovered in successful search for genetic processes in which chromosomal genes were transferred from one cell to another. We must thus account for the ability of F plasmid to transfer genes other than its own. Ability of F plasmid to transfer genes other than its own. This ability was understood when it becomes clear that the F plasmid could exist in another form in certain cells in addition to its autonomous existence as a separate plasmid. The F plasmid was found to be able to undergo recombination with the host chromosome. Undergo recombination with the host chromosome. And since both the molecules are circular, a single recombination event results in integration of the plasmid into the chromosome. The conversion of free F into integrated F is a rare event occurring in about 1 in 10 raised to 5 cells. These rare cells are called HFR cells or high frequency of recombination. HFR in the sense high frequency of recombination. Leading to HFR cells. Okay, so these HFR cells are there. The F plasmid can integrate into the chromosome at many different locations. So a variety of bacterial strains can be generated, each containing a single integrated F plasmid at different spots on the host genome. The molecular basis for the entry of the F plasmid into the chromosome at specific locations, IS sequences, was described earlier. So the F plasmid genes responsible for making the cell male are expressed in HFR cells. The HFR cells thus can mate and transfer DNA. When the integrated DNA in the HFR strain is nicked by the endonuclease and transfer of the DNA strand into the recipient cell begins, DNA from the host chromosome can also be transferred by virtue of its physical continuity with the fDNA. This transfer of host DNA into the recipient potentially can have profound genetic consequences. DNA from the donor is transferred at a rate of about 50 kb per minute. About 100 minutes are required for transfer of the entire bacterial chromosome. In most cases, however, the mating pairs are not stable enough for the entire chromosome to be transferred, and the cells break apart, leaving the recipient with a partial copy of F plasmid and a segment of the donor genome. Most often, therefore, only a subset of chromosomal gene is transferred because the origin of transfer is within the F genome. One part of the F plasmid is the first to enter the recipient, but the rest of the F plasmid will be last to enter. Therefore, complete F copy is not present in the recipient unless the entire chromosome is transferred. Complete F copy is not present in the recipient unless the entire chromosome is transferred. As a result, the recipients of most HFR transfers are not capable of being donors. As with autonomous F plasmid transfer, replication in both cells accompanies single-strand transfer. Left to its own devices, the pieces of chromosomal DNA transferred from an HFR donor has little chances of survival and propagation in the recipient. However, because of sequence homology with genes of the recipient chromosome, it can undergo recombination mediated by the homologous recombination system of E. coli. The recombination event can be detected by appropriate selection or screening. For example, the mating between certain HFR loop plus cells and HFR LU minus recipient. F minus LU plus cells can be detected. In the cells, the recombination events result in the replacement of LU minus genes with the incoming LU plus gene. So formation of mating pair. After that, the HFR chromosome is nicked in the integrated DNA sequence. Transfer of the donor chromosomes starting with the F sequence occurs into the female cell. Replacement DNA synthesis occurs in the donor HFR. Mating pairs usually separated before complete transfer occurs. In this case, after the C plus gene has been transferred, but before the D gene has entered the recipient cell. REC-A mediated recombination occurs between the transferred DNA and the host chromosome. Following chromosome replication, two cell types exist, one like the original F minus and second in which the B and C genes have been replaced. Then HFR transfer can be used to investigate gene location. HFR transfer can be used to investigate gene location. The analysis of many HFR strains revealed an important feature concerning transfer. Different HFR strains reproducibly transfer different bacterial genes at high frequency. For example, the hypothetical strain HFR1 might transfer genes A, B, C at a high frequency and genes X, Y, Z at a low frequency, while another strain HFR2 may transfer genes XYZ at a high frequency and genes ABC at a low frequency. This behavior is due to different insertion points for the F plasmid in the HFR donor's chromosome. Those bacterial genes oriented to one side of the ORI T transfer point are the first bacterial genes to enter the recipient and will be transferred at a higher frequency than genes further away from the ORI-T. Since genes are transferred in a linear order to the recipient in the HFR transfer, the time of transfer of a particular gene reflects its relative position of the bacterial chromosome to the origin of transfer. HFR strains, thus, are important tools for determining a map of the bacterial chromosome, that is, the order of linear array of genes on the chromosome can be established by determining the time of transfer of a particular gene relative to the transfer of other genes. The relative physical locations of the genes can be determined. Then F plasmids insert into specific locations. What determines the F plasmid insertion point? The F plasmid carries several special DNA sequence also present in several copies in the bacterial chromosome. The short insertion sequence, abbreviated as IS sequence, were described earlier. The transfer properties of most HFR strains can be understood as a consequence of recombination event between the IS sequence on the F plasmid and the homologous sequence on the chromosome. We have seen that the F plasmid can display two distinct lifestyles in a cell, either as a free plasmid or integrated into the chromosome to form an HFR. A third type of F plasmid has also been observed. This form, called F prime, results from an imprecise excision of F from the chromosome and a re-establishment in the plasmid form. The excision can also remove bacterial gene adjacent to F, which are then carried in a plasmid form. The excision itself is a result of recombination event between sequences within F and sequence outside. Many different F prime plasmids have been described. Some carry very large blocks of the bacterial chromosome. Like F plasmid, F prime plasmid retain the ability to transfer themselves to female recipients as long as they have not lost any of the genes necessary for transfer. F plasmid transfer results in the efficient reproduction of the associated chromosomal genes into the recipient. However, the genetic consequences are different from HFR transfer. After transfer of F minus, F prime, the F prime associated bacterial genes can be stably maintained by virtue of the ability of F plasmid replication origin to function. Thus, recipient F prime cells can carry two copies of certain genes, one of the F prime plasmid and one of the bacterial chromosome, and are genetically described as merodiploid, partially diploid, in contrast to the normal haploid state of a bacterial genome. Since F prime plasmids are much smaller than the bacterial chromosome, Their complete transfer usually occurs between mating pairs spontaneously break apart. Consequently, the female recipients are converted into F negative males. F negative plasmids are usually designated by the chromosomal genes they carry. For example, F prime lac pro carries the genes for lactose utilization and proline biosynthesis. Then plasmid DNA replication. As mentioned earlier, a fundamental property of bacterial plasmids is that they exist at characteristic copy number of cells. Plasmid copy numbers are usually expressed as a ratio of the number of plasmid molecules to the number of chromosomal copies per cell. Naturally occurring plasmid exist at a wide range of copy numbers. Certain plasmids such as X factor F or prophage of E. coli Ceras P1 exist at copy numbers of 1 to, while those of Cole even family exist at copy number of 20 to 30. Plasmids are generally dependent on host enzymatic machinery for their own replication. The Cole even plasmid, for example, carries no genetic information specifying replication related enzymes. It relies entirely on host components. Other plasmids may encode certain enzymes involved in particular steps of replication, but in no known case is plasmid completely independent of host replication enzymes. E. coli contain 3 different enzymes that catalyze DNA synthesis: DNA polymerase 1, 2, and 3. Most E. coli plasmids, as well as the E. coli chromosome itself, use DNA polymerase 3 as the central replication enzyme. An important exception is the Cole even plasmid family, in which RNA polymerase 1 plays a key replication role. Next, plasmid copy number. Plasmid copy number is regulated by controlling the frequency of initiation of DNA replication. Initiation of plasmid replication occurs at an ORI replication fork starting at ORI. Move either unidirectionally or bidirectionally around the circular plasmid. So, most plasmid replication forks are bidirectional. The Cole even plasmid, however, replicates using a unidirectional fork. Two questions of importance are: what special sequence of features of ORI enable it to serve as a replication starting point? How is the frequency of initiation events controlled so as to maintain a specific copy number? Many plasmid ORI sequences are organized similarly to ORI-C, the bacterial replication origin. A key functional feature of origins in general is that the DNA double helix must open in that region, permitting access to the bases by DNA polymerase and other enzymatic replication machinery. The site-specific opening of the helix is accomplished in several steps. The first step is the recognition of the ORI region by a specific DNA binding protein. For ORI-C of the bacterial host, the key protein involved in replication initiation is encoded by the DNA A gene. Many plasmids encode an analogous protein which is required specifically for their replication, called a rep protein. It plays a key role in initiation of plasmid replication. If the rep gene is missing or mutated, plasmid replication cannot occur from the plasmid origin. Moreover, a plasmid copy number is related to the intracellular level of the rep protein. The function of the rep protein is to recognize specific short RNA sequences at the plasmid ORI. At the ORI, other host encoded proteins interact with the bound rep protein to assemble a DNA replication enzymatic machine called a replisome. Once bound to the ORI, the replisome causes local unwinding of the helix, followed by RNA primer formation and replication of leading and lagging RNA strands, DNA strands. The specificity for assembling the replisome at ORI comes not from sequence-specific binding of the replication itself, but rather from an initial DNA sequence recognition by the rep protein, which direct subsequent replisome assembly at the ORI. So, based upon that knowledge, molecular biologists used initiative reasoning to speculate that intracellular levels of rep protein would be carefully regulated. In fact, plasmids have evolved highly sophisticated molecular mechanisms to control the production of this critical protein. These control mechanisms sense the concentration of plasmid DNA in the cell and regulate the production of the rep protein accordingly. When a plasmid is introduced into a cell, initiation of plasmid replication occurs quickly to establish the plasmid at that steady state copy number. This requires relatively high level expression of the plasmid rep gene. After the plasmid is established, rep protein levels must be controlled to prevent runaway plasmid replication, which can be lethal to the cell. The amount of active rep protein is controlled at the level of translation rather than by post-translational alteration. For example, the expression of rep genes of some plasmids, such as the rep A genes of the R factor R1, is controlled both at the transcriptional and post-translational levels by separate plasmid encoded gene products that repress rep gene transcription.